that, I guess I'll go ahead. With that, I'll go ahead and get started. I'm going to share my screen and we'll just get going here. All right, everybody. Well, thank you for joining here today. My name is Phillip Alberti, and I'm a research program manager in the Ellison Lab. And I'm here today to talk about using data to select compliant hemp cultivars. My contact information is on the screen. If you ever want to reach out, please do not hesitate to do so. So today we're going to talk about quite a few things. First of which, I think it's going to be nice to give kind of a general production overview on what hemp is, and then looking at how the different types of hemp are grown, which will kind of inform what I'm going to talk about our research efforts that are currently underway. So we're going to talk about production systems. We're going to talk about the current research that we have underway and the preliminary findings, and that is spelled wrong. We're going to talk about the Ellison Lab and the research efforts that are currently underway, and then opportunities for collaboration. So the first things first is to discuss, what is hemp and the differences between hemp and marijuana. So cannabis sativa is both hemp and marijuana. The only difference between the two is the amount of THC, or that psychoactive cannabinoid, that can be found in the plant. So if it's below 0.3% THC, it's considered to be hemp. If it's above that 0.3% THC level, it's considered to be marijuana. So really the distinction is a chemical one. They are the same plant, and in one day the plant could be considered hemp, and the next day in that that field it could be marijuana if those levels exceed 0.3%. So it's very important to understand that distinction. Hemp is primarily dioecious, although there are some monoecious varieties that are currently being developed, and it is an annual. But ultimately, what you need to know is that hemp can be grown for three things. It can be grown for food, or grain. It can be grown for fiber, and it can be grown for flour. But ultimately, the end use is going to determine the production practices. So if you look at the top left here, you can see that, for example, growing fiber type hemp, you want it to be very tall, very highly densely packed to allow those plants to grow very tall, have more competition. At the end of the day, though, no matter what type of hemp you are growing, the material needs to be compliant. So whether that's grain, fiber, or flour, all hemp needs to be considered to be compliant through your state, federal, or tribal regulatory bodies before you can harvest it. So this compliance testing is really just ensuring that your hemp tests below 0.3% THC, and that non-compliant hemp must be destroyed. It's important to note, too, that where you sample on the plant does matter. So THC, CBD, essentially cannabinoid concentration will decrease as you move down the plant canopy. And so following these guidelines that the regulatory bodies are going to follow for their sampling is very, very important. You'll be doing yourself a disservice to test lower portions of the plant, because the sampling agents will come out and test that more highly densely populated top part of the plant. So this is really where the crux of most of the research that's being done that I'm going to talk about today comes from, is this idea of compliance and determining compliant cultivars. So current state of hemp production is that hemp processing is still quite limited. This is across the board for grain, fiber, and CBD, despite this kind of boom that we saw in 2019, 2020 on the CBD or cannabinoid type hemp. Processing has been limited, and many growers took a hit and are still having issues finding processing relationships and establishing kind of that connection. So that's developing. Where we've seen an increase lately, at least, is in the grain and fiber processing, where there are estimated to be about 20 processors online in 2023, which is significantly more than we've had in the last 3 or 4 years. Regardless of all the processing, we're still figuring out how to grow hemp and to do it well. And so regional variety performance is limited, as well as best management practices for hemp. This is grain, fiber, and cannabinoid hemp. We have a long way to go. All this is to say that growing compliant hemp is a challenge, and you're going to see that with some of the data I present here shortly. And that seed certification for cannabis is in its very early stages, and in some cases feeling very non-existent, to say the least. So finding good sources of genetics and ones that are going to perform well agronomically from like a yield or a quality component, but then also be compliant with state, federal, tribal regulations is kind of this quagmire that we're navigating right now. So all of this led to the development of what is called the Midwestern Hemp Database. And this is an interactive display that allows growers, processors, regulators to access variety performance and agronomic performance of all of the trials that we have done across this kind of Midwestern group, including researchers from the University of Wisconsin, Michigan State University, Purdue University, and University of Illinois, Urbana-Champaign. And so all of the data from our agronomic trials and our variety trials go into the hemp database. This includes not just agronomic data, but cannabinoid development data, which is very important when making these selections. And the nice thing about it is all the data is currently available, and it's an interactive display, so you can kind of mess around with it to pick out the data that you want. And I'm going to show you that in a moment. But really, what's very important to understand is that all the data that comes into the database is coming either from university research trials, replicated trials, or grower cooperator trials who are working with us, as well as the Cultivar Check program, which is a series of grower cooperator variety trials. And we'll talk more about that. But all of this data gets put into the database and is available for the public. So how does this thing work and how can it be used? And so this is the house for the hemp database. And so what I wanted to demonstrate here is that we provide the research reports from each of the universities, and all of that information gets put in here. But then you can access kind of this interactive display that allows you to look at different types of hemp, whether it's grain, fiber, or cannabinoid type hemp. You can look at a specific genetics provider, and then all of the information for that variety or for those selections that you've made will be updated in real time. And this is true for both cannabinoid, hemp, and grain and fiber. And we include data on cannabinoid performance, agronomic performance, summary statistics, and even some figures, images to really kind of flesh this out. So by the numbers, over the last 3 years, we've had over 200 grower cooperators across the 4-state region send us samples or contribute to this in some manner. We've had over 250 different varieties be tested, and over 2,200 samples, which is a considerable amount when each of those tests costs about $75 commercial value. And what we found over those 3 years is that 28% of all samples that were submitted to the database are non-compliant, which is a problem. That's more than a quarter of the samples. And if we were a regulatory agency, would have to recommend that, or prescribe essentially that 28% of those lots would have to be destroyed. And so this really, really puts into perspective that we have a clear need for variety recommendations and harvest guidelines. Something I'm very proud of, the Midwestern Hemp Database back in 2020, when they reopened the public comment period for hemp, submitted data from the hemp database to address several contested topics, and data from the hemp database was used to support rule changes for several of those contested topics, including increasing harvest windows to give growers more time to harvest their material, and raising the negligence threshold for growers to essentially allow them a little bit more wiggle room before potentially losing licenses over noncompliance as this new industry is kind of developing. So very proud as a researcher, science informing policy in a positive way is one of the most important things you can dream of. So this has been a great and a very proud moment for our team. So now moving into, the specifics of the research that we do and kind of the differences in production systems, this is really where I think I'm really excited to talk about this. So I think it's important to understand these differences in how the hemp is actually grown. So when we grow hemp grain, hemp is actually an oil seed. Grain gets thrown around quite a bit, but it is an oil seed. This is very similar to small grain production, where we're using seed stock and we're drilling it in, or using some sort of broadcast with light incorporation or a modified planter. But typically a grain drill is going to be your best bet when you're growing grain or fiber hemp. And our seeding rate recommendations, as you can see, range from about 25 to 30 pounds per acre. But we like to use pure live seed as a recommendation for seeding rates. And I'm going to talk more about the data from our trials and what that's shown here shortly. Harvest time. Grain hemp is extremely challenging to harvest. It's very similar to things like carinata or canola, camelina, where shattering is definitely an issue, and so harvest timing is critical. Even in your best day, you're really only going to be harvesting hemp grain at about 70 to 80% maturity to avoid losses from shattering. And this is going to be using some sort of grain or a draper header, which is going to be important to mitigate losses due to shattering and really be able to harvest those grain heads. What I'll say too is if you look here, it's important that we try to mitigate fiber wrapping in grain hemp harvesting. And so only the top portions of these plants where the grain heads are clipped. The remaining material is often left in the field and can be chopped or baled eventually for what we call a dual purpose crop, but primarily that material is broken down in the field or, tilled and incorporated. It's a very high carbon material, so keep that in mind as well. And then drying of the grain after harvest is also a challenge, more challenges. There's forced air is the only way we can dry down hemp grain quickly before spoiling. Using heat will cause the oils to oxidize and then spoil and risk losing your crop. And yields from our variety trials in the region show that between 600 to 1,800 pounds an acre for grain is expected, with averages around 1,200 pounds per acre. For some of our higher performing varieties. Now, fiber hemp is very similar. The only differences I can really tell you about the establishment of fiber hemp is involving, in some cases, the fertility, but mostly in terms of seeding rate. So if grain hemp, and I'm going to go back to grain hemp, seeding rates are about 25 to 30 pounds per acre and about 15 to 25 plants per square foot. For pure live seed, we're doubling that for a fiber plot. And again, that's because we want the hemp to grow, have high competition, grow very tall, to have pencil width type hemp stalks, which allows for easy retting and subsequent processing after the fiber has been harvested. So again, really the only difference between establishing grain hemp and fiber hemp is you're doubling your seeding rates. Hemp is chopped at anthesis or flowering. It's allowed to ret in the field for about 2 to 3 weeks and then can be baled up before sending off for further processing. And yields for fiber hemp typically range between 1 and 3 tons per acre, but we have seen some varieties that produce as much as 5, and we can talk more about that later. Really, so again, the importance here in the harvesting is that hemp fiber is actually harvested or chopped at anthesis, so right in between that kind of late vegetative, early flowering stage, and then the oil seed is naturally combined at harvest, so we are talking September in the fall when that grain will be harvested. Fiber chopping typically occurs in July, allowing for the retting process and baling to occur sometime in August. So now that we know a little bit more about these production systems, we're going to talk about the research that we've been building off of the last few years. So we recently were awarded the USDA NIFASAC grant for canola and hemp to focus on hemp production. This is building off just general preliminary work that we did that was not grant funded over the last few years. Our collective group of researchers across the Midwest basically asked for donations and handouts to allow us to get some preliminary data to write a grant, and also grants just for hemp really weren't available over the last few years at such a scale that would allow us to do work like this. And so we're fortunate that we were able to get the grant and look to continue this work on a more replicated basis for the next 3 years. And so information on that is on the slide here, where we're going to be taking replicated trials across the 4 states at each of our respective research stations. And then also adding a grower cooperator component, 5 growers in each of those states to grow strip trials of those varieties that are in our university station trials, to build out this network of understanding how varieties are performing, using row crop producers who are using equipment at scale to really evaluate the performance, and then hopefully connect with some fiber processing to start developing out this network on a regional scale. So I don't have much data to share from our preliminary work, other than to say that this was what it looked like last year at our University of Illinois plot, where I was at the last 4 years. And so we looked at things like variety evaluations, germplasm and evaluation and improvement of feral hemp feral hemp lines, and then also looking at agronomic performance. So planting dates, row spacing and seeding rates, fertility and tillage practices, all of which we're evaluating, have been evaluating, and will continue to do so as part of our larger efforts moving forward. But I do have a few things I would like to share from our preliminary work about the data that we've collected and how it can be used to inform selections. So I think it's very important to understand, again, that because of the state of hemp seed certifications across the country and across most of the world, actually, seed quality for hemp is a little iffy at best. We can look to our state regulators or state seed certifying agencies who have kind of certified seed, or we can look to AOSCA, the Association of Seed Certifying Agencies. But across the board, what we see is that there are differences significantly in seed quality, seed size, and weight across hemp cultivars. And so it's going to be really important to focus on germination and seed quality testing. Because of the lack of certification right now, we just can't bank on what we're getting. And so we perform independent seed testing on all the seed that we get as part of our trials. And just to demonstrate these differences here, you can look at the amount of seeds per pound and the range across genetics that we have entered into our trials, where we can look at a variety such as Jinma, which has about 15,000 seeds per pound, versus something like A2, which has 43,000 seeds per pound. So this is very important because the seed size changes dramatically how much seed you're going to put in the ground. And if we're using seeding rate recommendations, it might not be the most accurate in terms of what are we getting for stand establishment. And so this is where pure live seed really comes into play when we're looking at both the seeds per pound and the standard germination test results to figure out a true seeding rate that will achieve our target population density. And so you can just see again here between the seeds per pound and the germination test, they're widely variable. And so we have to account for this in our trials. And so an example of this is, in 2019, we used to think about seeding rates of grain in 15 to 25 pounds of seed per acre, but our mindset has changed based off of seeding rate studies to really start looking at, again, this targeted pure live seed rate. And the same goes for fiber, where we were looking at seeding rates, and now we're looking at targeted populations of pure live seed. And so an example of this, specific example, would be if we assume that we are growing a grain variety and we want to establish this at 20 pounds of seed per acre, looking at two different varieties, factoring in the seeds per pound and the germination rates, you can see that the amount of plants per square foot that we get is extremely variable. It's going to change a lot based off of the seed quality. And so we can kind of course correct in our trials for poor seed quality by looking at this targeted pure live seed population density. So this is one of the things I really like to push growers who are growing hemp to look at, is to see how these are going to factor into their management decisions. So how can we use the database itself? One is to look for certified or eligible hemp strains. These are varieties that may have been certified by AOSCA or comparable certifying agencies. They could be specific countries. They could be, places here in the States, seed improvements, and in various states, for example, like Colorado has some that have been approved. But look here, because this will give us at least a starting point for varieties that are performing uniformly with some sort of expectation of how they should look, perform, and those qualities, the baseline for seed quality that we would expect from a commercial line. So look for certified hemp seed. The problem with certified hemp seed is it does not take into account cannabinoid development. And so this is where the hemp database is very useful, is to couple this seed certification with its performance via our trials. Look for things that are certified, are going to perform well, then look to see if they're compliant based off of our data. And last but not least, look for high-performing hemp strains using each of your regional variety trials. So we put all of these three things together because they're all very important. They're all asking very different questions. And it's important to understand that as this hemp space is starting to really develop. So again, look for eligible or certified for eligibility varieties. look for compliant hemp strains via the database, and then look for high-performing strains via your regional agronomic trials, which give you a really good, indication of their performance. Any questions so far on the grain and fiber and kind of where we're at and where we're going? Okay. So now I'm going to talk about high cannabinoid hemp, and honestly, probably for the remainder of my time, this is what I will be talking about. I will say that the hemp database factors heavily into everything that we do. So there's a reason why I talked about that first, because it's going to play a role in what I'm talking about in just a moment here. But I do want to give kind of an overview on hemp, high cannabinoid hemp, or hemp for flower and or biomass for extraction. And so this is very much like a specialty crop or a vegetable crop. If we're talking about grain and fiber as a commercial row crop, this is much more of a labor-intensive specialty crop to grow high cannabinoid hemp, whether it be for THC, CBD, or any of the other cannabinoids that are currently emerging on the market. This is typically how it's done for outdoor production, where we are looking at transplants and clones as the primary form of establishment, and some day-neutral varieties that are produced more like a row crop. They may be planted from seed, but typically we're talking about transplants or clones on about 6 feet row spacing, anywhere from 4 to 6 feet typically. So this is a lower planting density, and we're looking at larger individual plants. Okay. So the target population here, we're looking at about 1,200 plants per acre, versus, again, grain and fiber, where we're talking about pounds of seed per acre, and there's thousands and thousands of seeds per pound. So this is a very different thing. The harvest time for cannabinoid hemp is in September, early October typically, and is done by hand. And it really depends on whether or not this material is premium flower, where they'll go through the field and really look at the best material and harvest that. And then the rest of the material will get harvested, ground up, and sent off for biomass for extraction into tinctures and gummies and things of that nature. So there's two kind of avenues that this can really go. But again, ultimately, this needs to be compliant for it to be harvested, and high cannabinoid hemp provides the largest challenge for producing compliant hemp. And so if we consider the issues that we've had with determining varieties are compliant, this led to the development of the Cultivar Check program. And remember, Cultivar Check program is one of the pieces of the places where the data comes from that goes into the database. And so really what we've done here is we look at all of our variety entries, and we've made a selection of ones that we feel would be good potential for the region. This is based off of grower ratings. This is based off of our own experience, but it's also, most importantly, based off of the data from the database. So which varieties have shown high potential are the ones that we would like to evaluate further. And so we include those into this Cultivar Check program. We send growers across the Midwest subsets of 4 to 5 of these varieties. So we get about 6 to 8 site years annually per variety entry. And then those varieties are evaluated not just at the university research stations, but at all these locations across the Midwest, of which we had 32 last year. And we test the varieties for germination, uniformity, and overall performance. But primarily, the biggest thing that we take is flower samples from each of the varieties at different times throughout the growing season. This is 3, 5, and 7 weeks. So we can figure out how cannabinoids are accumulating in the plant, which varieties have a good chance of being compliant, which don't, and then factor that in with performance agronomically to make true variety recommendations. All the data that I'm going to share with you is available on the Emerging Crops webpage, where we released our 2023 Cultivar Check program report from the 2022 growing season. And one thing I'll mention too is just, it's a very neat tool that allows us to do this, is we manage this trial using SeedLinked, which is a tool that allows us to evaluate the varieties in our trial, but set up these mini trials and connect with growers. So just a brief overview of how this works. If you ever run trials like this and are looking to do something similar, we can create a trial. So let's just say check number 7, or Cultivar Check number 1, excuse me. We can pick the varieties we would like to have in that trial. We can determine which of the qualitative metrics we would like to evaluate the varieties for. And then we can take pictures, we can communicate with the growers, we can get planting date, flowering date, all that information, and we use this as a way to validate the data and also to validate when varieties start to flower. So each variety from each grower must submit a sample, picture, and a date, get verified by one of our collaborators before we consider that variety to be flowering and can start a sampling protocol. So results from this trial have really just kind of showed that uniformity of flowering within and among genetics or varieties, is extremely variable due to the lack of seed certification standards. This means that, within a seed lot, we have varieties that are flowering unevenly at different times of the year, the growing season, or across, very long periods. So some may be across long periods, some may be very tight. And so really what we're looking for is taking advantage of uniform varieties that look the same and perform the same and have tighter flowering windows, so we have less of an issue with potential compliance. Because you can imagine if you have one variety, but many different phenotypes in the field, all expressing different flowering and cannabinoid development, how can you expect as a grower to truly know what's in your field? And that's really where this issue comes in. So seed certification is everywhere, is a problem everywhere. And we're seeing this in our replicated trials along with the grower trials. What's really important is that most CBD dominant cultivars are going to exceed 0.3% THC at some point throughout the growing season. So if this red line is the regulatory threshold, any sample above that line would be a non-compliant sample. The data presented here is presented in such a way that for each variety at each time point, the value is an average. And I can talk more about why we decided to show the information in that way. Basically because the impact of location and flowering periods were not significant, so we took them as averages across all data points. But this allows us to see which genetics have more potential to go hot and when they do. So, for example, some varieties like Buffalo Soldier may take a very long time before it exceeds that threshold, where things like Suver Haze, for example, is basically non-compliant shortly after week 3. So this is very important for growers trying to time their harvest, if they can know that on average, most of these genetics are going to be non-compliant between 5 and 7 weeks after harvest. Total CBD. CBD production is what drives profit potential, essentially, in hemp. And so total CBD plateaus for certain cultivars and will change based off of a genetic, but it becomes very important to understand the relationship between when CBD is maximized and when THC becomes non-compliant, because it's a tightrope walk that you have to make to try to get the most profitability out of your field while reducing risk for non-compliance. And lastly, on this, THC and CBD have been shown to be very stable throughout flowering periods and across environments, and show a very linear relationship in terms of as CBD increases, total THC increases, and this is just given the metabolic pathway by which THC and CBD are produced. But really what this means is that growers can utilize this to establish a relationship between CBD and THC, which is about 25-to-1, which is what we recommend growers who are looking to grow high cannabinoid hemp target for, is looking at the CBD to THC ratio of that genetic, which will give you a good idea of its profit potential versus risk for noncompliance. So what do we do across both years of data collection from the Cultivar Check program over 21 and 22? We have decided to create harvest timelines, because this really answer the question of, can we use confidence intervals for cannabinoid development to develop a harvest timeline for a genetic? And so at each different time point, week 3, week 5, and week 7, across all samples, so in this case we had 14, this is the total THC lower confidence level and the upper confidence level at 95%. And we did this for all genetics entered into the trial over the last 2 years, and with the hope of kind of, again, creating this harvest timeline. So varieties that are highlighted in red were non-compliant at 95% confidence before week 3. Varieties in yellow were compliant through week 3. Varieties in orange were compliant through week 5, and green varieties were compliant through week 7. And so this allows us to kind of color code these varieties and give growers an idea of when they might need to start sampling for cannabinoids or be concerned based off of variety performance across all of our trials. And so we did this for every variety into the program, giving growers some sort of idea of how they're performing. What I'll say is that we are continuing this project in 2023, 24, and potentially 2025. And so we're going to add more genetics to the pool and continue to test them. Varieties that receive poor performance are removed from the program, and we continue to bring in fresh blood to test them and see which varieties are performing well. I apologize for the clarity of this image. It's coming from our report, but just wanted to demonstrate, the amount of data that we're collecting for these varieties. So the last thing I'll say about the check program and the data that we collected is this is utilizing grower cooperators. And so we make some concessions in terms of the way that we collect data. We do a lot of qualitative data collection, which mitigates the labor that a grower would have to do. We have to be respectful of their time and understand that they are doing this in many cases kind of pro bono. So we decided to pick very few but targeted ratings that we would like growers to collect on a 1 to 5 scale. We provide them with a key for what each value would mean, and then we would we publish this data here in terms of summaries across all varieties. We understand that there's a lot of issues with differences in skill level, differences in environmental conditions, and so cannabinoid development does not seem to change in response to stresses. What does seem to change is overall performance. And so while we do put this data out here, when making selections, we really push for utilizing our university station variety reports to look at things like yields and germination, but we still provide this data for the growers because we have it and it is substantial. We've also been able to establish kind of a timeline for flowering where we can pick early or later maturing varieties to give growers some sort of choice as well. So again, the idea is still the same for utilizing the hemp database to make selections. Look for seed certification from AOSCA or comparable agencies, whether it's in your state or in the region. Look at the hemp database to look at variety performance to make from a cannabinoid development standpoint to see which varieties are going to have a better chance of being compliant. And then from there, look to our university station trials to make a selection based off of agronomic performance. And I think between all 3 of these things, you put yourself into a really good position to make an informed selection. And this is across the board for both hemp and for grain and fiber hemp and for cannabinoid hemp. One thing I did not mention, I'd like to tell you is that we have both a cannabinoid hemp database and a grain and fiber database that we keep separate for the time being, but the rules stay the same. They're interactive. All the data that we have is available. We're working on a way to combine them in such a way that you can just filter out the data, but that'll come soon. So last few minutes before I kick this over to Derrick I just wanted to talk a little bit about some of the other work that's currently been being done in the Ellison Lab. I am the program manager for Dr. Shelby Ellison, and as you see here, our current focus is preserving, characterizing, and utilizing genetic diversity in alternative crops to meet the needs of Wisconsin farmers. And so I'm going to talk a little bit about each of these things very briefly before allowing Derrick, Dr. Grunwald, to talk about his work. So some of the work that we've been conducting, Dr. Adamola Aina has been collecting, characterizing, and preserving hemp germplasm across the region, basically looking for feral hemp collections, whether this is legacy or wild, legacy hemp from World War II or wild hemp that's been around for a long time. We are looking to classify and categorize these collections as part of a larger breeding effort. So if you're aware of feral populations and want to contribute to this national project, let us know. We're happy to go out there and explore and look for these varieties or these feral populations to contribute to this larger effort. But we're really looking at traits that regulate CBD and THC production, as well as seed banking, so we have this for future breeding efforts. You can contact myself or Dr. Aina if you have any information on these sites. Grace Connolly is looking at evaluating ecosystem and agrisystem services from companion crops and hemp to see which companion crops do or do not grow well with hemp, to evaluate if they have an impact on yield or quality of both the companion crop, which in some case could be harvested for value added, or for the hemp itself. So I thought that was pretty fascinating. We talk a lot about the draw of hemp for pollinators, but there's also, detrimental insects as well. So seeing how these two different types of plants interplay with each other is fascinating. You can contact Grace, and her information is there available as well. Sean Kim is looking to understand anthocyanin production in cannabis. So this purple pigmentation, which is caused by anthocyanin accumulation, is a desirable trait in the cannabis industry and is growing in more and more popularity, it seems. And so Sean is really looking to understand which of the factors influence anthocyanin production in cannabis. Things like UV exposure or cool temperatures, to start understanding the pathways responsible for this accumulation and where within the plant. Is it in the floral tissues? Is it in the stem? Is it in the leaves? And what's causing this, and what conditions are causing it to increase or decrease that regulation? Before we move on to Dr. Grunwald, I just want to point out the collaboration efforts that we have and opportunities. So whether you're looking to grow grain and fiber hemp or CBD type hemp, we have a project for you. As part of the Supplemental and Alternative Crops Grant, we are looking for grower cooperators to grow replicated strips of these varieties for us, anywhere between 1 and 3 acres. We provide the seed, we provide a $500 stipend, and if the material is compliant, you may keep it at the end of the season. Hopefully you have an outlet for it. But that is what we're looking for. So if you know of any experienced growers or looking to contribute, please let me know. You can scan that QR code or reach out to me or find information on our website. With regards to the high cannabinoid hemp, we have the Cultivar Check program. This is funded via the SARE partnership grant, of which we were awarded again and will continue for the next few years. And this is looking at CBD and CBG type hemp. We're looking for growers to grow replicated 4 to 5 varieties, about 25 plants per variety. And we're looking for experienced growers. We send the seed, we pay for the cost of testing, we pay for the cost of shipping. It does not replace compliance testing through your state agencies, but it's a great way to trial new varieties before taking them on at scale on your farm. So if you're looking to get involved or interested, want to know more, please contact me. I'd be happy to talk to you more about this. And lastly, all this information is available on our new Emerging Crops website, which the team has done such a great job of putting together. All the information from feral hemp collections to disease diagnostics and getting involved with our trials, all that information is available there. Huge shout out to our team. I may have said I or me a lot. It's a we across the board for all of these trials. Extension, growers, researchers on campus, private labs who have donated time and money to allow us to do this work, like Rock River, who this project would not be have taken place without their generosity in providing discounted testing rates. So thank you to our seed suppliers. This has been a great collaboration, and we look forward to keeping it going. And that's our team. Before I came to Wisconsin, I was at Illinois the last 4 years, and I hadn't met any of the folks on our team throughout the development of this project over the course of the pandemic. And so up until about 5 months ago, I hadn't met anybody, and we were working on this together. So it was a great experience to see and meet everybody, and just looking forward to keeping this work going. So I'm going to stop right there, and I'm going to kick this over to Dr. Grunwald, and I'll take your questions after. Thank you.
All right. So everyone hear me, see me?
Yep, you're good. If you want to put it in presentation mode, then we will.
Slideshow. Does this work?
Yep, looks good.
All right, great. So as Phil mentioned, I'm a postdoctoral researcher in Shelby Ellison's group. My background is in plant pathology, so I'm just sort of moving forward with plant pathology and hemp. My presentation will be sort of briefer than Phil's, so let's get started. Oh no, there we go. So in general, when thinking about like plant disease management, we're thinking about sort of different strategies to avoid plant diseases, including like avoidance, eradication, so destroying the material, exclusion, protection, using resistant varieties. So I'm interested to see where this moves in the future, sort of curative therapy. So these things can be like heat treatment for getting rid of viroids, for example. However, in order to test the efficacy of any of these management principles, you need methods to detect plant diseases. And that's sort of where my research comes in. I'll just briefly introduce the one that I've sort of been using. So in general, in order to detect multiple diseases at one time, you can either run conventional PCRs. So in this case, you would have a single sample that would be separated into 4 or 5, however many different reactions. So in this case, I'm showing 4, and each reaction would have a different primer set. So in this case, it'd be 4 different reactions. Versus in a multiplex PCR, you would be detecting the same things and a single reaction that contains all the primer sets of interest. So this has the benefit of sort of reducing the amount of materials used, or requires additional optimization to make sure that none of the primer sets, for example, interact with each other, and you can detect everything fairly at the same rates. So sort of, I'm going to skip over all the how we did it part. In general, when we were doing this multiplex PCR, we decided on detection of 3 viruses in a viroid. So these include Cannabis sativa mitovirus 1, citrus yellow vein associated virus, beet curlytop virus, and hop latent viroid. So we determined that we wanted to work with these, as previous research has shown these to be at least present in hemp in Wisconsin and Colorado production systems. And we'd sort of been collaborating with a few Colorado labs in order to get materials that we could then easily test. So it was more a test of convenience, but these are all viruses that are of interest to Western growers, as well as a few of them that could be of interest to sort of Wisconsin production of note, hop latent viroid. So I'm just showing here a gel image that shows the blank, which is B, positive control for the various viruses or viroid, which would just make sure that our reaction is working, and samples 3 through 7 are just a selection of a few different independent hemp samples that were supplied to us. And so you can see sort of the relative position of the different viruses. So this is mitovirus on top, around 500, around 400 base pairs for the citrus yellow vein associated, 300 for hop latent viroid, maybe 200 for beet curlytop, and around 100 for an internal control, just to make sure that we were able to extract RNA and synthesize it successfully and to see DNA from the various samples. So what I'm showing here is, in general, just we can detect all the viruses that our multiplex set out to detect and plant samples. So for example, sample 3 has a mitovirus, hop latent viroid, and sample 4 has mitovirus, citrus yellow vein associated virus, and beet curlytop virus. I should note that this is part of a manuscript that was submitted, so that's forthcoming. So the main results from the manuscript is from our Colorado samples. We found this mitovirus is prevalent in around 90% of tested samples across 2 years. Co-infection ranged from 8% to 20% of tested samples across 2 years. So we tested samples in 2019 and 2020. And we've shown that this technique is pretty effective in determining the presence of virus, but it should be combined with orthogonal techniques for confirmation. So I don't want to be out here and say that the multiplex is the only technique you should be using for determining viral presence, especially if you want to know the sort of viral titer. A multiplex PCR would not be sufficient to tell you that. Moving forward, so this is just if you have any interest or know of people who might be of help here. I'm looking to develop novel diagnostic methods for other diseases of interest. So these include alfalfa mosaic virus and tobacco streak virus, so AMV and TSV. In particular, we're interested to know whether or not there are any hemp-specific TSV strains out there? And if so, can we design particular diagnostic techniques specific to that hemp strain? We're working towards developing surveying disease prevalence in Wisconsin production systems. So we're going to be screening again for podaviruses, tospaviruses, TSV, and AMV. This would be using immunological methods, so like ELISA. And then lastly, sort of a more large scale or more broad thing of interest is to know what the disease priority areas are of growers. And so sort of conducting surveys in order to figure out what the priority areas are for further research and how we can adapt sort of a diagnostic scheme or research into diseases to more tailor to those needs. That's sort of all I presented or had ready to go. So if there are any questions about that, please let me know.